Some months ago, So Yu found orange mould in his bathroom. Orange. It was unexpected and beautiful. He wondered what other colours are invisible to us. So he started looking for more. From the samples he collects, he grows an ever-increasing variety of moulds. And he makes these creatures sing. Even though singing is not the best term for now. So Yu is a Taiwanese communication student. Like many others around the world, he learnt what he needed to know for his project on internet, or by asking a friend. This is his interactive map of Taipei. This work of art makes the viewer realise the abundance of creatures in the seemingly life-empty cityscape. He is far from being the only one to engage in biology in his free time. For instance, in the same city, the Taiwanese group Taiwan BioArt is dedicated to biology as an art medium.
And one night I was really bored and I couldn't sleep. So I started to search on the internet for the word biology and art. Biology as a media is unique. If you are prepared for 10 issues which might happen during a bio art project, you will definitely get the 11th.
Taiwan BioArt members gather every Wednesday. They talk about the latest blockbuster, or if it's possible to make yogurt from vagina bacteria. Thanks to biology and lab protocols, they tell stories and ask questions. If someone injects human DNA in a plant after death, would that be reincarnation? What does modern synthetic biology tell us about our society's vision of life? Is it possible to recharge phones with our own blood?
I draw blood from human and use that to make a chemical battery to generate electricity. We do workshops here. I think what we found is that people in Taiwan, they are not as afraid of genetic engineering as we thought. A lot of the news are about how dangerous genetic engineered food are, and we thought that if we do a genetic engineered project outside of the lab, people would get really shocked and think we should be jailed or something. We were prepared for that, but people were actually quite amused. Which was out of expectation. And because we were doing the transformation in front of them, I think we sort of demystify genetic engineering, and they were like, oh, this is not too hard, I can do it too.
People who engage in biology without a degree say that they are doing DIY biology. Do-it-yourself biology is primarily based on the principle of open science. Thomas Mboa, PhD in microbiology, has a nice definition of it.
Open science, I can define it in three combined ways. Open science is not limited to technology, but technology is a great tool on open science. Open science is open to all the actors without consideration of academic level, intellectual skills or technical skill. Open science is also mainly collaboration, open collaboration, free, without any barriers or financial barrier, geographical barriers, and whatever.
Do-it-yourself biology is more precisely embodying the very different aspects of hacking, like open data, knowledge sharing and citizen science, but also hedonism, rebellion, passion, community spirit, individualism, entrepreneurship, and a mistrust towards bureaucracy. Pierre Padilla from the Peruvian biomaker community is not pleased with this definition. Too intellectual, not practical enough. So we could say it is rather a biotechnological social movement in which individuals, communities and small organisations study biology and life science using the same methods as traditional research institutions. But then Morgan Meyer, sociologist and specialist of the DIY bio movement, finds it narrow. Maybe it's the act to engage in biology outside of a traditional laboratory. Some people involved in the movement would find this terrible. So let us say that DIY biology is a little bit of all these definitions, and maybe even more. As it is practiced worldwide, encapsulating the aims, means and motivations is like trying to describe a loved one. The best thing we can do is to give but a glimpse of what it can be, and let everyone make their own opinion. Moreover, the name itself is not clearly defined. Some call it DIY biology, but for others it's rather biohacking or even garage biology. But biohacking also refers to the improvement of one's physical and mental abilities through habits scientifically deemed healthy. It also refers to the grinder community, people who seek to improve their bodies using implants or chemicals. This is confusing. One could say that's because it's a new thing. Everyone seems to have a different version. Some would say that the starting point was in 2010, opening year of the first physical DIY biology lab, GenSpace in New York. Others are arguing that it was 2 years earlier, in 2008, with the creation of DIYbio.org, the first global online DIY biology community. Some are claiming that biohacker is a term that has been coined, in fact, in 1988, in an article titled Playing God in Your Basement. As if the definition wasn't blurry enough, a few people are saying that the discovery of the antibiotic penicillin in the 20th century is itself a form of DIY biology. Maria works as an engineer in a biotechnology company. During her spare time, she dedicates countless hours to a DIY bio community.
So I was originally a volunteer at BioCurious when we opened over 7 years ago. I showed up. I was very interested in what was happening here. I was involved in biotech at the time. I don't have a background in biology, but I wanted to learn more. You know, I've heard from some friends that there was going to be this biohacking space started. It would be a place where people could be able to do their own experiments, and I thought that sounded really exciting. You know, I'm from when in the 90s there was a lot of computer clubs where people were getting together and, you know, the 2600 clubs, and you were trying out code and you were trying repurposing things and you were building things from spare computer parts. And so the idea of doing that with biology really intrigued me. So BioCurious was founded primarily by Ari Gentry, who at the time was trying to do a cancer startup. Her and her co-founder looked around Silicon Valley, and they couldn't find anything less than $3,000 to $4,000 a month for 6 feet of bench space. And that was far outside the realm of what they could do, and they had this great idea for cancer research, and what they ended up doing was starting a lab in her garage. So out of that, she said, you know, there's a real need for people who want to do basic research to just be able to work at an inexpensive place to pursue research. And that has been a great unmet need. The original funding money was actually crowdsourced, which was very new at the time. So we raised $35,000 that way, just through crowdfunding, and we started. you assume we'd start, it would be amazing, but what was the most interesting to me is that from getting started, no one knew what to do with the space. People were just looking at each other going, well, what do we do now? What projects? I don't know. Do you know what we should do? No one really knew what to do. And out of that, after about 3 or 4 months, we said, okay, this space is not, getting used. How do we get momentum back? And community-based science is what we came up with. And that has become a cornerstone of what BioCurious is and is my passion here. When a lot of people talk about community-based science or community-driven science, a lot of those projects tend to be what I consider accounting type projects, where people go out and perhaps they'll take surveys or they'll count how many butterflies are in their backyard, or they'll go observe things or take pictures. But what they aren't doing is necessarily making new discoveries, and they aren't participating in the data side or the design side of the research project as much. So they're being used as almost interns for someone else's research. And we wanted to turn that on its head. So what we did is we brainstormed some projects, and the community thought of the projects.
The bachelor mix, which is going to be this work, the primer mix, which Maria is getting, and then That's interesting.
So this is the start of our lab. We want it to be fun, a little bit whimsical.
BioCurious is one of the 55 DIY biocommunity labs registered at DIYbiosphere.com in 2018. Representing all those communities would be quite a challenge, so we can assess their diversity through examples. BioCurious has its own building in the Silicon Valley. It's one of the oldest DIY biolabs. A few kilometres away, Counterculture Lab is part of a shared space for non-profit local initiatives. BioFoundry in Sydney is located in a startup hub, thus aiming to be a stepping stone for young entrepreneurs. Biomaker has its quarter in Lima University. It focuses on building a DIY Bio-Latin American network. Kumasi Hive, a Ghanaian open source innovation centre, is about to start its own DIY bio community.
Which is like the most traditional technique of transgenics.
Those labs are what sociologists call a third place. It's not a home, it's not work, it's a third place. This creates an atmosphere where people can meet, engage and express themselves without constraints. A space where money is mostly a commodity, and where you can talk about changing the world. Exactly like a bar, but with more science and more focused conversations.
I think we might all be going to hell.
I'm not going to hell.
Yeah, you are.
It's a scary place. I don't want to go there.
Spaces like these are essential for DIY biologists. It eases the process of scheduling, debating, learning and simply doing. It allows the members to feel legitimate and committed. Also, it's easier to sleep when your bedroom is not hosting the next experiment. It can be difficult to find a suitable place, though. It needs not only to be affordable and accessible, but also safe enough to experiment. Lots of initiatives died for not finding one. An empty web page is a grave. To fill the community space and to engage in biology, we need a variety of things. Mostly weirdly shaped glasses and machinery.
So the main machines we use are the incubator for growing things, refrigerators for keeping things cold, PCR machines for amplifying out DNA, centrifuge for spinning things, 3D printer for printing things, UV transilluminators for looking at DNA and dyed DNA, we've got a couple of those. Autoclave downstairs is absolutely necessary for almost all biological things. RO water machine, of course, is also very important so that we can have very pure water. And more refrigerators. We've also got a -80 downstairs, which is valuable for keeping things more than a year.
There's an issue though. Machines are really expensive. For instance, this rather odd plexiglass box with a wire at each end is called an electrophoresis box. And this is nothing compared to more sophisticated machines. These costs have been making it nearly impossible to engage in biology without extensive funds. Do-it-yourself bio-communities can gather some resources through crash courses, membership fees, occasional grants and crowdfunding. Yet the amount they usually get would make any professional scientist sceptical about what kind of science they can do.
Wells are a bit further away from that end. Yeah.
This does not stop DIY biologists.
I've started doing the same kind of thing myself, where I make friends with university contacts, and when old equipment is throwing out, we're the first people they call, and they say, hey, do you need a new bacterial shaker? And I say yes, and they say, well, it's broken in this way, and we decide whether or not we want to bother trying to fix it up. And if it seems like it's something which we can fix up, we fix it up. If we overestimate ourselves, it goes into storage with the thousands of other pieces of machinery that we didn't manage to fix. For the most part, we've been gathering stuff over the last 5 years, and as things are fixed and made functional, we'll move them into the lab and start using them, as long as it's not going to take up too much space. Except for the auto-pipetting station, which is literally just a useless waste of space and will never work.
Sometimes this is not enough. So we can buy an electrophoresis box and maybe eat cup noodles for an undetermined number of weeks. Or you can make one. That's what Alex did. And there it is, for $50 Australian, even after an unsuccessful first attempt. Do-it-yourself biologists create more and more options to hack the costs to engage in biology. We can buy a PCR machine to build on our own, entirely open source, costing up to 5 times less than the professional ones. Looking in your kitchen might be enough. A pressure cooker makes an excellent autoclave. And with some thinking, a 270-fold microscope can be built with some cardboard and an LED. As for atomic microscopes, or any next-gen expensive machines, it is highly improbable to find them in any DIY biolabs. But basic machinery is more than enough to perform quality work. To experiment with biology, we also need consumables. Chemicals of all sorts, enzymes and other microscopic tools. It can be quite difficult to get our hands on those more or less transparent liquids. Some components are not available to amateurs due to their dangerous nature. Some are just way too expensive. And in some countries, you might get arrested for importing ingredients known to be involved in cocaine production. Do-it-yourself biologists think wide to get what they need for experiments. If an enzyme is too expensive, we can nicely ask the small quantity we need to the company that sells it and see what happens. If a chemical is too dangerous, an older protocol that nobody uses anymore might not require it. All these solutions imply long hours of thinking, networking and browsing the internet. A small price to pay to avoid getting in serious debt. The most important ingredient for a successful experiment is people. A potential DIY biologist has 2 resources available: time, to spend hours learning, debating and experimenting, and curiosity, that is to say the reason to spend this time doing science instead of looking at cute cats on internet.
We've got some members who come in once every week or once every couple of weeks. We've got other members who are in here every day. Some of our members, for example, will be doing mushroom growing, so they'll need to come in each time they need to refresh their generation. For example, Kevin's got his spider here, so he has to feed this spider every time she gets hungry. She's just sitting up there. A lot of it comes down to emotional bandwidth and whether people are actually have spare energy left over after they finish their work and study to go on and engage in things. A lot of our members are fascinated and excited and want to get involved, but then find themselves completely exhausted at the end of a university day.
Come on in. Hi. Anyone wants to make some
Do-it-yourself biology aims to be inclusive, as there are no formal barriers to participate. One does not need a degree, or even money.
So anyway,
As of skills, well...
Look, I got a Bachelor of Science from a great institution, the University of Texas. When I left, I didn't know how to do science. I had a lot of vocabulary, a lot of theoretical knowledge. Here, the first thing you learn is practice. You get the theory later. And so it's really flipping the process on its head. Theory is great, but what it does is it boxes in possibility. And so somebody who doesn't have theoretical knowledge may try something that somebody who has theoretical knowledge wouldn't. They said, no, that's impossible. It's not worth the time.
Nowadays, most of the DIY biology communities have a core group of about 2 to 10 motivated people. Most of them are scientists, engineers, artists or students, people with pre-existing interest in science and with enough time and energy to engage in a community. Given that DIY bio-community labs are fairly recent, we can hope that more people will join the movement. More people means different backgrounds, different skills and different frames of mind. Diversity, that is the main and unquantifiable riches of a DIY bio-community.
It's really important for us to have a balance of all different types of voices, all different types of backgrounds, because it's a core belief that when you incorporate all these different types of people, all it does is like create a nice ferment for innovation. So novel ideas coming together in novel ways, right? Universities and science in general can be quite siloed or separated by discipline. Those walls, when we look at existence and life, don't actually exist. And so you might think that you have stumbled on a solution, but since your solution is in isolation, it won't actually have impact, or it won't actually work, or nobody might even care. Innovation basically is taking ideas that exist and connecting them in a novel way. And ideas exist everywhere.
Do-it-yourself biology aims to bring more people around the table of biotechnology, not only on a local scale, but also on a global scale.
The difference between Latin American initiative than other initiative from United States of Europe is that we didn't want to start with synthetic biology. We wanted to make basic things. Like, for example, build your own centrifuge, your own incubator, your basic tools that you need. If you go to any university, you will find that students have to share the equipment or the tools, different tools of the lab, because you don't have enough tools there. And that's why I think scientists, researchers, or other people see that it's a good initiative.
Do-it-yourself biology communities are not closed among themselves. On the contrary, they seek interaction with other actors through their projects. It can be universities, as Pierre explains. It can also be companies.
We've been working on real vegan cheese for many years now. We're finally getting some headway there, and that project could have easily become a startup company. It still could. But we want to create the technology to have yeast express the proteins that make cheese, to make something that is chemically cheese, but doesn't come from an animal. And the reason for that project is specifically, if we can create this technology, make it open source or license it at very low cost, you'll get larger market spread of it than if a single company owns the patent and really restricts how it can be used. And overall with that project, we'd like to reduce the use of animals and also reduce the greenhouse gas effect of people eating cheese. One of the things we have discovered from that is that the vegan community is very excited about the project because it's not a for-profit. So because we aren't an industry, because we are seen as outsiders, they're actually more accepting of it. And because it turns out they really want good cheese. So their fear of GMOs is superseded by their desire for cheese. So we may have found that, key point. So again, getting people to understand GMOs and the fact that they can be used for good. It's not just Monsanto using it.
It's a really tough thing about my class.
Do-it-yourself biology communities also seek interaction with government.
People are engaged in Western context because it's like an hobbies. But in Africa, it's different. People are engaged in this movement to survive, to find a job, to solve a particular problem. If the state is not able or is not interested to do this kind of work, we are helping to the state because our first interest is to give our population a well-being. So that is our first interest. And if the government fail to do that, we can help the government. It's not an opposition or alternative government inside the state. No, it's just to help the government to do his job. Business in open science. Sometimes you have the promoters.
Open Science Hardware Africa, or OSH Africa, is a convention about open science and open access innovation. The first edition was organised in Kumasi, Ghana in April 2018. It gathered a wide variety of tinkerers, about 80 Africans and 20 people from around the world. The first event of the sort on the African continent.
We don't have the same challenges like in US or in France or in Latin America. That is why it was so important to organize this event in order to see or to rise up this kind of issue, this kind of challenge, particular to our context. There are many hospitals in rural zones, for example, in my country and in many countries in Africa, which don't have even the small equipment to run small diagnosis to patients. So biohacking can be helpful at this stage.
For instance, some cardboard, wood and strings can replace a $1,000 worth machine. It has recently been discovered that this hand-powered centrifuge can spin blood samples up to 120,000 million round per minute. This seemingly easy solution allows remote village dwellers to diagnose themselves for anemia. Much better than a 10-hour ride to the nearest hospital.
Africa Open Science and Hardware is a starting point of a big conversation about this topic, the open science and hardware, in our context, and try to improve the life of our local community here in Africa. We are open to the world and we are trying to build together a better Africa.
While being local-oriented, these groups form an informal global network based on their similar interests, passions, and a sense of community.
So I'm going to buy both of those from
Local communities are where things happen, but they could barely exist without the global network. The global network is a powerful tool, but it is only useful if it helps local communities. Through Facebook pages, meetups, Twitter or mail lists, biohackers from all over the world are in constant contact, helping one another.
Just grab that.
In Latin America, a group called Syntec Bio ties different communities of the continent together. Don is its founder.
So the only way to do biohacking in Latin America was by doing it together, and that's how Syntec Bio was born. Organizations that, for example, are in Germany and say, we want to do this in Brazil. Does anybody would like to help? And then the people of Brazil say, let's do it together. You can bring this, we can do that. So it really, really broke down the barriers. The last activity we did was we created a book that helps you to build biohacker spaces with really low resources for all the links that are in the internet, and was really a good example of working together because everybody have Google Docs and we're working at the same time in documents. Somebody was curating the documents. We have people from the United States like Maria from BioCurious helping us to make sure that the English version was really well written in that language. And then we have people from Africa that just say, we like what you did. Can we just translate it to French and use it here in Africa? Yeah, sure, just do it. It's open. And I'm gonna say the truth. I don't know much about how things work there, and that's actually how the network works. It's an uncentralized network. I don't want to be the guy saying what people need to do because I have no fucking idea.
This open source philosophy that we have, you can replicate anything, you can modify, you can propose new projects that maybe you can help other countries. Like, for example, we always talk about some common things that we have Latin America and Africa, like infectious disease, like malaria, dengue, Zika. We have a project where we are developing a basic incubator for detecting infectious disease. And all this machine, it's open source. If this kind of diagnosis works here, I think it could be easier to replicate in other countries.
We're all dedicated to open source.
Open source.
Open source.
Open source.
Open source is about sharing a content, like a software or a book, and allowing anyone to use and modify it for free. In other terms, it's knowledge sharing. And one of the fundamental resources to engage in biology is knowledge. Ranging from knowing how to unravel the mysteries of the rose DNA to knowing that you shouldn't mix this with this.
I think the most important thing that I've learned here at BioFoundry is that if I don't know how to do it, I can probably teach myself how to do it. There's a great deal of expecting you to reach out to your mentor and get them to explain to you exactly what you need to do when you don't understand the problem at university. And I think BioFoundry really challenged me, because I didn't have those resources on hand the entire time, really challenged me to be able to find ways of finding my own solutions and engage in the online community myself. So, and it also taught me that there's always someone available with the answer somewhere in the world. So if you have a good enough community around you, you can always reach out to those people and ask them what their opinion is, what their experience is, and hopefully do the same for them one day when they ask you the same question.
Chloramphenicol. At 100 degrees, it is stable for 5 hours. Wow, okay, well that's totally sweet then. Some antibiotics are a lot more sensitive.
Most people don't understand how political science is. It is very, very political. It's not just because you have to apply to a government organization like the National Institute of Health to fund your research. There are certain topics that are not researched because they're in some way taboo. There is established dogma in science. And so what we're doing here in the same way is political. And it's political because we're providing access to anybody. So we're saying you don't need a degree from a university to do biotechnology. You don't need a PhD to run a research project. You don't have to keep secret everything that you're doing in order to have a product or an idea come to market. There are different business models that exist.
Through the use of technology such as 3D printers, you can get a virtual thing that a lovely stranger sent from the other side of the world and translate it into a physical object with no particular skill and just a €300 investment. This allows more than printing cute dinosaurs. It decentralizes production of more and more objects, such as prosthetic limbs, while keeping low costs thanks to open source. We just did the full circle between software and hardware, data and objects. It is a virtuous circle. Low-cost hardware allows more people to engage, increasing innovation, allowing better and more accessible hardware. Talking about ideas and innovation is, most of the time, leading to intellectual property considerations. Most do-it-yourself biology communities simply encourage their members to share what they are doing. As long as they are following the safety rules, members can do their own project, eventually patent it, and start a company. The goal is not to block innovation, but to free it. It appears that entrepreneurs might be bolder if the test phase of their project is not costing them all of their savings. For instance, Pili, a company producing coloured inks thanks to bacteria, was created in a DIY biology lab. In pop culture, labs are white and immaculate for a reason. They are the kingdom of science, great mistress of modernism, heir of high, inaccessible knowledge. Do-it-yourself biology labs are different. Truth be told, engaging in biology can be dangerous. Not so much for the ill-intended mad scientists.
Make this instead. And biology's like, actually, I'm going to continue making my own things, or I'm just going to implode so that the rest of my brothers and sisters don't die. Yeah.
Engaging in biology can actually be dangerous, but mostly for the experimenter. That's why every new member of a DIY biolab has to pass a security exam. And they will get banned if they are not following the safety rules.
Why biology is so frightening is because of pandemics and the idea of the potential for bad actors in biology, for controlling biology, the idea of controlling what we are, who we are. It can be frightening, but what you do is you want to bring them into the conversation. You want to bring people into your space. You want to show them the kind of projects you're working on. And I have never had somebody come in here and see what we're working on and be anything other than happy and excited. And that means that we've had everything from, we are in our third year of having the military come in from the Pentagon, including, brigadier generals. Take a look at our space. We have a yearly, once-a-year meetup with them to evaluate DIY Bio for safety. We've had, the FBI, various government agencies, they come in pretty regularly, and we have nothing to hide. DIY biospaces need to be aware of the media, of PR, and of getting ahead of stories. And if anyone is interested in more on that, I'm always happy to help.
Do-it-yourself biolabs are making biosafety institutions acknowledge them. They ask and obtain accreditation for specific manipulations depending on the country's laws and regulations. And the communities are following those guidelines for a very simple reason. They don't want their labs to shut down. For a government, do-it-yourself biology can be quite scary. However, according to some, enthusiasts will always find a way to get their hand on what they need to engage in biology. So, in case DIY Bio is declared illicit, it would most probably go underground, burying with itself innovations opportunities. While, if it is authorised, the communities can strive, and the government can monitor it. Working with DIY Bio could also help policymakers by pointing some out-of-date biosafety regulations, or on the contrary, showing that an aspect for security must be reinforced.
I think there's a lot of technology that when it was created, it was like a taboo. So one of those examples is the Makerpiece. The Makerpiece was created, it took almost 10 years for the MDs to use it, because they thought it was unnatural to keep people alive that should be dead. So you have a technology that was born, and you have 10 years actually of gap in which people was dying even having the technology because the society couldn't believe that you should keep them alive. Well, all the biotech we think right now is crazy. In maybe 20 years, it's gonna be the standards for us because it shows value. So for me, about technology is between the balance between the risk and the value. And obviously, every technology that we use has a risk, So imagine how dangerous a car is. Would you stop using it?
So, is do-it-yourself biology going to change the world? Is it going to revolutionize our relationship to knowledge and science? Is it going to spawn a generation of carefree mad scientists?
Don't put so much pressure on us, man. Doesn't mean we're not trying to change things, but we'll see.
I'm working with anti-aging. I have worked in machine learning a little bit. I have worked in synthetic biology, in biohacking. And there's the problem of the hype. So the hype is interesting because it kind of pulls a lot of people interest into trying to solve problems and technologies. But at the same moment, the hype can be really dangerous in terms of how the public understand what we're doing. So it's interesting to be really excited about it, but I think also at the same time you have to be really careful about what you say in public when you are a person that works with this and know what he's talking about, to try to be as clear as possible with people so they don't get, for example, scared without any reason, or try to actually do damage into a technology that by itself has no problem, but it depends on how you use it. So I always, when I see a lot of really, really excited people, I think it's amazing. But I think also it means, okay, let's talk about it, let's study a little bit, let's see what is going on, let's try to see the pros and cons, because everything will have also a downside, so we need to evaluate those too.
Depending on which newspaper you're reading, biohackers might save the day or destroy the planet. As of 2018, DIY biologists are not considered serious actors in the research world. Lots of projects have been abandoned. Lots of communities ceased. Most labs are still struggling with finances. A vast majority of DIY biologists are upper middle class, working in a field related to science.
We're still really early. Watching over the last 5 years, DIY Bio just being, a handful of labs, maybe 7 labs all around the world, to I think that we're at like 50 or 60 labs that are on all continents except for Antarctica. And that is really amazing. We've also seen that these spaces are communicating together on a regular basis, that they're starting movements that are local but are touching on their local community and local institutions.
Do-it-yourself biocommunities are creating local networks between people, universities and government. They promote alternative models in terms of intellectual properties and knowledge economy. They create cheap hardware useful in many countries. And they engage in bolder and bolder projects every year.
One of our other larger projects here that is probably the most scientifically challenging project we've ever attempted at BioCurious is our new cuttlefish project. The reason I don't know that many labs would attempt something like this, but we just happen to have the right amount of expertise here to tackle it, or we don't know that we shouldn't tackle it, is this idea to sequence the dwarf cuttlefish. Very few cephalopods have been sequenced. So we said, this is too cool. Let's sequence it de novo, because it's never been sequenced before, so there's no reference genome to refer to. Reassemble that genome, do the RNA sequencing. The scope of the project, cost-wise and resource-wise, is enormous. We'd like to create some cell lines for research and make them open source. So they've never done a cell line for any mollusk or cephalopod. They've never figured out how to. People have tried. We have an idea for a CRISPR gene knockout using CRISPR-Cas9 that we think will allow us to do that. A lot of people have told us this is bigger than what they think a lab like us can do, but we've been chunking away at it. And again, these are the kinds of revolutionary research that, unless you had researchers who are in a lot of different areas, they might not think of these ideas, or they might not get the grants to do them because they're so out there. But we can just try them.
They don't fear failure because they know that failure is part of science. They know that negative results are results. They know we often can learn more from our mistakes than from our successes. Well, They also don't feel failure because they will not lose their jobs if the project crumbles. As Marie says, do-it-yourself biology is nowadays at a child stage. Baby stage was 8 years ago, and the creation of the first physical labs. Teen stage could be the first replicable scientific results, or the success of a project like real vegan cheese. Child stage is now, this weird and fascinating movement, without a defined form, but with already huge ideas and means to make them real. Some US universities are already trying to replicate DIY biology. One could wonder about how they will deal with intellectual property of what happens in their artificial DIY biolabs. A lot of interrogations are still floating around it. Some labs have achieved financial equilibrium, but how about the long run? Will people from other professions join the movement? Will institutions, such as universities or research institutes, restrain or encourage DIY biology? Will biohackers stay true to their starting intentions? Can they achieve the same reproducibility and statistical proofs we see in a traditional lab? What would happen if... Well, we'll have to wait and see.